So here we're just gonna go through the structure, and then we head into the mass flow head pressure flow hypothesis. Now the composition of the phloem, we know that we have sieve tube elements, right? So usually we call them elongated cells. They have generally thin walls with perforated plates along the ends, right? So this is generally the structure of the phloem, right? They have small amounts of protoplasm rather, my bad, protoplasm within them, right? See, showing the fact that they are still somewhat alive, but because the protoplasm does not contain all the organelles necessary, then we're gonna need to have a companion cell, right? So they contain pores, right? The sieve plates contain different pores that allows for the movement of the sugars and organic solutes, right? And photosynthetes to travel through, right? And general instruction of the sieve tube elements is for the movement of the organic nutrients, right? The sap from the photosynthetic tissues, which are the source, to the various parts of the plant, which is the sink, or which are the sinks, right? But the companion cells specifically, they're small nucleated cells supported that they have nucleus, right? Closely associated with the sieve tube element, right? So they provide all the metabolic support, right? They have all the cytoplasmic contents, right? They maintain the cytoplasmic contents, right? Of the sieve tube elements specifically. They include the energy production. We're going to look at why they need energy to be produced, right? And they provide some amount of protein synthesis. So we're looking at the fact that the protein, not the protein, the companion cell itself, right, really supplies all of these cellular processes for the sieve tube member or the sieve tube element, which just has a simple protoplasm, right? And we have the movement of these things into the sieve tube element via a plasmodesmata. All right, so they're connected via plasmodesmata. The phloem parenchyma, right? So there are other interspersed cells among the sieve tube elements, right, and the companion cells. So they're similar to the xylem parenchyma, in which they would store starch and other types of nutrients, right, for the support and maintenance, right, of the phloem. Okay. All right. So this is not an important thing to note per se, but within the tube, specifically the sieve plates, right, we're gonna have like cytoplasmic strands in it that also allows for more movement of water. We're gonna have slime bodies, right? So usually the younger the plant, the more slime bodies exist, right? They're not important to know, right? But that's why when you actually like cut open like small plants and stuff like green plants, right? Some of them are actually really, really slimy, right? And then they would have the sticky sap. Right? Just something interesting, right? We don't really note the existence of the callus and the slime bodies, right? We have phloem fibers as well, so they're long, narrow stretches. So these are the fibers between them, right? They are comprised of lignin and cellulose, and that helps us provide mechanical structure to the phloem. So in the xylem themselves, the walls themselves are lignified, right? And they have supporting xylem fibers. But phloem now, since they're not lignified, they are going to need lignified cells, right? Composed of cellulose and all sorts of strong materials in order to support the structure of the phloem the same way. So the sieve tube elements themselves are not lignified, but the phloem fibers are. Alright? Some notable features, we have the phloem sap, right? So it's a nutrient-rich solution, right? Filled with primarily sugars such as sucrose, right? Amino acids, you have hormones and all these different types of organic compounds are filled within the phloem sap, right? So they act as a medium for transport, right? So a good amount of it is water as well. So it acts as a medium of transport for these organic nutrients and photosynthesis, to produce during the photosynthesis, right? The non-photosynthetic parts, right? So basically, it facilitates movement of nutrients from the source to the sink, right? So the sap there, right, is the, let's say, the plasma, right, through which everything travels, right? The medium through which everything travels, right? And then we have the pressure flow mechanism, right? So it's a mechanism by which the phloem sap moves through the plant right, driven by different hydrostatic pressures, right, between source and sink, right. So it allows for the bulk flow of translocation, right. So that's another thing that the phloem supports, right. And the pressure flow mechanism, or the hypothesis itself, is the next big thing we're gonna be looking at, right. So we're gonna be looking at this more in depth when we get there. So structure and function relationships, just going over these. We know that the sieve tube elements, right, are specialized for the efficient movement of these nutrients, right? And we know that the companion cells are there to ensure active transport of sugars against concentration gradients and to provide metabolic support for the sieve tube elements. We know that to be true, right? We also have support and protection. These are via the phloem fibers and the parenchyma. So the phloem parenchyma would store starch and different nutrients for the maintenance and repair of the phloem tissue, while the phloem fibers would be lignified and contain cellulose in order to support the structure, right, itself, the structural support, right, of the phloem tissue. We also have metabolic support, right? Well, I mean, I spoke about the metabolic support already, right, from the companion cells. That shouldn't be much of an issue there, right? So those are the general structure and relation functions, right? Nutrient transport and structure and protection, and support and protection rather. And we need to know the different structures that do these different functions, because that is what biology is. At the end of the day, when you strip biology bare, it's always structure and function. That's why I always separate biology slides and stuff like that into what the something look like and what it do. That is all biology really is about, right? So use our knowledge of structure and function to answer questions, all right? So bulk flow as well is another important function of the phloem, right? It drives the movement of the phloem sap through the source, right? From the source to the sink, right? So it ensures the continuous supply of nutrients to the plant, right? And that is what we're going to be looking at. We're going to be looking at phloem loading and then the entire hypothesis, right? And then we're going to be looking at the end of it there. We're going to call it the end there for the first topic once we get through this. Okay? So phloem loading, right? Loading really occurs within areas that have a high photosynthetic value, right? So usually loading occurs against a concentration gradient, meaning that sugars move from an area of lower concentration to an area of higher concentration, right? Generally something like that. So usually loading occurs within photosynthetic organs, right? For example, the leaves, right? So we have sugar production in the mesophyll cells, right? So we know that photosystem 1, photosystem 2 do all of its nice electron photo excitation, all sort of stuff, and the NADPH goes into the stroma, and we have Rubisco, right, championing all this photosynthesis, yada 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 yada, right? When the sugars are now produced via photosynthesis, right, we're going to have, right, accumulation of these sugars, right, in the chloroplast of the mesophyll cells, right? So we're gonna have the accumulation, right, of them, right, in the cells, right, and cytoplasm specifically, right? What's gonna happen here now? Active sugar transport into the companion cells now, right? Once synthesized sugars will be actively transported from the mesophyll cells into adjacent companion cells located next to the sieve tube elements, right? Remember, there's a vascular bundle in the leaves, right? So the sugars will be moving towards the vascular bundle, right? So the active transport process involves the specific transporter proteins embedded in the plasma membrane of the mesophyll cells, right? And utilize ATP, right, to move the sugars against their concentration gradient. So in this case, they're moving from a high concentration to a lower concentration, right? So they're moving across that gradient there, against that gradient there. So up here we could see this, right? So we have the mesophyll cells, right, moving, right, well, creating this proton gradient, right, which will be used to produce ATP, right, to move all these sugars into the sieve tube, right? So we're gonna have the sugars moving from the mesophyll cells, right, into the companion cells, and the companion cells will facilitate this movement via active transport into the sieve tube elements. And this is what loading really entails. This is a major factor of loading, All right. So we have that there. Everything is fine here.
Sir, you can go over that again, please.
All right. So in simplicity, right, what we're looking at now is the fact that the mesophyll cells produce sugars, right, and we need to get these sugars into the sieve tube elements, right. So we're going to have the active transport by way of the companion cell. Remember, the C2 elements don't have these mechanisms, right? Therefore, the companion cells will have to do these metabolic processes. The companion cells, right, will use a proton gradient, right, to set make ATP synthase to produce ATP, and this ATP would be used, right, to be a form of energy, right, that is used to actually move the sugars against their concentration gradients, right, into the companion cells, and from the companion cells into the seed tube elements. So what we're looking at is the active transport of sugars, right, from the mesophyll, right, to the apoplast, from the apoplast to the companion cells, and from the companion cells to the seed tube elements. But usually, it's generally from the sugar producing cell to the companion cell to the seed tube element via active transport. Right? We have there. Shouldn't be a problem. Is that better? All right. So
Wait, the apoplast, is it an actual part of the thing or is it like a pathway or a route?
it's a pathway. So you remember symplast and apoplast pathway?
Yeah, I do, but when you mention it like that, I thought it was like an actual thing
Okay, it's because why I mention
rather
it like
than a
that.
pathway.
Okay, why I mention it like that is because we're going to be moving through the intracellular membrane space, right? And there is an apoplastic movement. That's why this diagram here has apoplast. So it moves into the membrane. It moves into the intermembrane space, and then into the companion cell and into the seed tube element, right? So that is really what is really happening. But some diagrams just show it moving directly from the cell to the companion cell, neutrophil cell to the companion cell, which is not exactly true. But you get what I'm saying. So it's not I wouldn't per se say it's an actual thing, but it's truly what's happening, right? So we have that there. So the sugar movement into the sieve tube elements now, right? We're just looking at it a little bit better now. We're gonna have the movement of the sugars through the plasmodesmata via connecting channels, right? So the plasmodesmata are the connecting channels, and the movement of the companion cells to the sieve tube elements, right, occur passively, right, driven by the concentration gradient, right? So looking at that. So in this case now, we're looking at the movement of sugars via active transport into companion cells, and then through the plasmodesmata, right, we have the movement of these sugars into the seed tube elements, right? So it's generally what I was explaining before, right? So active transport into the companion cell, and then through plasmodesmata, right, into the seed tube members, right? The establishment of the concentration gradients, really, right? What we're generally looking at here is the fact that when sugar accumulates within the sieve tube elements, we're going to have a concentration gradient being set up, right? Their concentration of the sugar in the sieve tube elements will increase, right? So looking at that first, right? So the amount of sugar in the sieve tube element is more than what's outside of it, right? And that creates a gradient between the source tissues, which are the leaves, right, and the sieve tube elements, right? So this concentration gradient drives the movements of sugars along the phloem from source to sink tissues, where sugar is needed for growth, storage, and our metabolism, right? So we're gonna look at that. So that concentration gradient is established there when more sugar is in the sieve tube elements than within the source, right? So we have that there. Therefore, it will cause sugar from moving will cause sugar moving in the seed elements to move from the source itself, right, to the sink. We're gonna be looking at that through the pressure flow hypothesis and how exactly that works, right? So sugar loading, right? We spoke about sugar loading already, right? We're gonna have the active transport of sucrose. I'm gonna be referring to it as sucrose or sugars, right? Into the companion cells, right? And then the passive flow of them, right, via plasmodesmata into the phloem, right? So we're gonna have that translocation. We're gonna have some amount of translocation there, right? We're gonna have water uptake due to osmosis. So we're gonna look at how the phloem and the xylem combined, right, create this pressure flow thing, right? So the high concentration of sugars in the sieve tube elements lowers the water potential in the phloem. And we spoke about the components of water potential before, right? As a result, we're gonna have water moving into the phloem from adjacent xylem, right? That makes sense, right? Since it's
Wait, sir, wait. Where say like it has thick but it not process.
okay, so remember, when we have high solute concentration, right, that will lower the water potential. And the water potential moves from high water potential to water moves from high water potential to lower water potential, right? So here, there's way more solute than water. Therefore, water would move from the lower concentration or high water potential to a to a lower concentration here, right? The high concentration over here to a lower concentration, or from a higher water potential to a lower water potential. So because the water potential in the phloem, right, in the sieve elements, right, are so low, it causes, right, water to move from the xylem to the phloem. So it drags water from the xylem to the phloem, right? And these are usually within source areas. So it creates a positive pressure or turgor pressure within the phloem sieve tubes, right? So when sucrose is in high concentration, water moves from xylem to phloem. Full stop. That is what we're looking at, right? And that pressure gradient and mass flow starts to come into contact now, right? So the positive pressure or the turgor pressure that is generated from what I just discussed, right, within the sieve tubes, right, create this pressure gradient between source and sink. So what we're saying, is that there's a higher concentration of solute up here where the source is, yes? Everyone can see that, right? And then there's a lower concentration of sucrose or any type of solute close to the sink, right? So therefore, that is a gradient. So the closer you are to the source, the higher the solute concentration, the lower the water potential. The closer you are to the sink, the lower the solute concentration, right? The greater the water potential. Alright? So what we're gonna have here now, water is gonna be leaving from the phloem, right, in the sinks. And water is going to be entering the phloem at the source. And that causes an entire cycle to occur. So let me use the writing and prevent the pen, right? So it's a cycle. So it goes around in a cycle, right? It's an entire cycle. So the water is going to leave from the phloem at the sink, right? Go to the source and it's gonna push the solute through translocation. That's what we call translocation. The movement of nutrients in the phloem, that is called translocation. So we're gonna have the translocation of solute from the source to the sink. From areas of high solute concentration to areas of low solute concentration. This is simple diffusion, right? But we can't call it simple diffusion because it's moving over such a large distance in such a complex way. We must understand it through the pressure flow hypothesis, right? Which is difference in water potential that causes turgor pressure, or the mass flow hypothesis, which is difference within the concentration, right, of the solute, right? So the pressure gradient, we look at the pressure gradient as the water changes, right, and produces turgor pressure at the source, and then the pressure will reduce as we continue to the sink. Mass flow now, sugar dissolved in the water flow from areas of high pressure to areas of lower pressure, right? So there's a high turgor pressure at the source and there's a low turgor pressure at the sink. So we have the movement from high pressure to low pressure, right? And then the mass flow of sap is driven by that pressure gradient established by the differences in solute concentration and water potential. So that is what the main thing here is talking about. That is generally what the hypothesis is. Let me pause. Are there any questions? So think about it a little bit and see if you can find any questions.
Sir,
Mm-hmm.
question. How does I know you kind of touched on it, but how does active transport play a role? Because I'm always confused with in the loading process. Because them
Okay.
always active transport play a role, but like two books are saying different things, and my teacher said something different, so I'm kind of confused.
Okay, so let me provide like this. From my understanding, right? Remember, this is a hypothesis. Alright. So notice that a theory and it's a hypothesis, right? So um let me fix that. That does not make sense right there. Alright. So let me just use the laser pointer. You see where I'm pointing here where the arrows move from the mesophyll cells to the companion cells?
Yes, sir.
This is generally done due to active transport, right? Okay.
Oh, so this,
Here. So notice the red balls are sucrose, right?
yeah,
So we know that more sucrose is in the companion cell than here, right? So we're moving it
it's
against a
weird.
concentration gradient. So it's active transport.
You know why I thought that was weird to say? Because I was saying that, I mean, I place where it produced should I have more, but I guess I don't know.
No, that's true. That's completely true, right? But the movement across, especially the apoplast, right, that would be through active transport. It makes sense, right? After a while, looking at a specific point in time, right, there will be more solute within the mesophyll cell rather than the companion cell, right? So that would be probably facilitated diffusion occurring there, right? But after a while, right, there's gonna be a continuous movement of sugars, right? So as sugar is produced here, it is moved within the companion cell, right? So there's gonna be a higher concentration. If you could think about the area as well, companion cells are much smaller than that of the mesophyll cell, right? So there will be a higher concentration due to the smaller volume, right? Therefore, active transport would need to play a role. Don't know if you understand it through that perspective.
Yeah, I kind of get it. I guess this is something we have to just know the facts and just, well, know the hypothesis and not question it too much.
Well, for the purpose of the examination, right? Let me just say that for the purpose of the examination, right? Because we had some arguments about it before in the past, right? Whether or not the hypothesis makes sense, right? But have a looking at this, right? So here's where the active transport takes place, and then here's through facilitated movement through the plasmodesmata, and then active transport would happen at the sink. We're gonna look at that. Alright? So with the unloading now, right, we're gonna have the movement from the sieve tube into the companion cell and now into the sink. So for example, a fruit where it would be stored, into a root where it would be stored, or into any type of cells that are growing, wherever the sucrose is needed, that's gonna happen, right? So the sink tissues are where sugar is needed for growth, storage, or any type of metabolism, right? So we're gonna have water reabsorption. So the sugars are unloaded, the concentration of the solutes in the sieve tube elements decreases, raising the water potential. Therefore, water is now reabsorbed into the xylem, reducing the pressure in the phloem sieve tubes, right? So we have that there. So water. So what we're gonna have here now is generally facilitated diffusion here. So um, sucrose is just gonna be passively moving through where it needs to be stored. Okay, we're gonna have that.
But sir, is it safe to say that you have both facilitated diffusion and active transport taking place at both instances?
Well, active transport. Sure. Wait. Okay. So in different periods now, we're gonna have facilitated and active. So you could say both.
Yeah, because like right here, you're going to have high concentration in the companion cell
Mm-hmm.
moving into the sink where there's less.
Mm-hmm.
So you would say it is moving down its concentration gradient. But after a while, when you have most of them in the sink, then it would revert if it's active, if it's down its concentration gradient, right?
Yeah. If it's against, it's
Yeah,
active.
so then you'd have both of them then.
Okay. So usually in developing things, right? So like developing fruit, it has not much sugar, so it'd be facilitated, and then as it starts to ripen, well, not ripen, We'll look at ethylene when we talk about hormonal control. But as it starts to be filled with sugars now, then active transfer would be the best bet, right? So we're gonna have a mixture of both. As the concentration changes and as the movement that we want to happen, right, either goes with the concentration gradient or against the concentration gradient, right? The recycling of water and sugar, so we know that the water would be reabsorbed to the xylem, right? And some sugars may also be reloaded into the phloem for redistribution to other parts of the plant, right? So we have that there. So that's really what's happening here, right? So this is a summation, sum up of everything. You get high hydrostatic pressure or high turgor pressure or high pressure overall at the source and the low pressure at the sink. Just going through it. Alright, so have a look at this diagram, and if there are any more questions, let me know. I'm gonna give you guys a couple seconds to look at it. Or screenshot it or whatever if you want. Have a look. So read through each point. Point 1, 2, 3, 4, and 5. What's happening? Alright? So point 1, loading at this into the phloem. Point 2, water enters into the phloem, right? Point 3, the translocation of sucrose. Point 4, the unloading of sucrose into the sink. And point 5, water returns to the xylem. This is a hypothesis. It's really simple when you look at it for a good while. It gets much simpler when you look at it. Alright? Alright. So, are we moving on?
Wait, sir, at 2.
At 2, what's happening at 2?
So you load your sucrose
Mm-hmm.
and into your phloem, and you have your water potential decreasing
Yes.
right at the top. You have your water moving in, right?
Mm-hmm.
And that would have been what caused. Okay, I see now. Thank you.
Okay, no problem. Alright, lovely. So, this is topic 1